Hello, my name is Jason Lutz, and I'm going to talk to you about purine and pyrimidine metabolism, Chapter 33. Hello, my name is Jason Lutz. I'm a molecular biotechnology major here up at Washburn University. I'm going to be talking about Chapter 33, the metabolism of purines and pyrimidines. And so what are purines and pyrimidines? These are the bases to our nucleotides, so they make up DNA and RNA. To begin with here, we're going to be looking at the anabolism of purines and pyrimidines. So when we look at metabolism, there's always two main overarching sections. We have anabolism and catabolism. And anabolism is where we are building up a molecule, and catabolism is where we're breaking a molecule back down. And the anabolism of purines and pyrimidines, one of the key things with this is we actually don't need to ingest these in our diet. Most organisms synthesize purines and pyrimidines on their own, and so they don't need to get that from their diet. However, there are a few protozoa that do actually parasitize nucleotides. This is also heavily controlled by feedback regulation. One of the big things we're going to look at with this is a molecule that we call PRPP, or phosphoribosyl pyrophosphate. This is the backbone by which our nucleotide rings, our nucleotide bases, are going to be put onto the molecule. This is made of a ribose sugar that's coming from our pentose pathway, and the pyrophosphate is actually added on to the 2' carbon here. I wanted to take a quick look at this carbon itself, as this is the difference between DNA and RNA as well. The 2' carbon in DNA has a just single proton on it, a hydrogen atom, where in RNA it has a hydroxide. And actually during the synthesis, it will continue to have a hydroxide, if not a pyrophosphate group on there during the synthesis process, and then later be converted to a deoxy nucleoside rather than a ribonucleoside. So when we look at the anabolism of purines and pyrimidines, there are two main pathways through which their anabolism occurs, and that is either through de novo synthesis, so starting from scratch, or from a salvage pathway. De novo synthesis can be a little bit different from purines and pyrimidines as we look here. De novo synthesis for purines can be awfully expensive on the body, and so salvage pathways are preferred for purines, whereas pyrimidines typically are de novo synthesized, and they don't have as many salvage pathways for them. That PRPP molecule that we were looking at before, the backbone, is really essential on getting this started, as this is the molecule that starts our purine synthesis, and it does have a feedback where it's going to increase synthesis of pyrimidines. So across all nucleosides, PRPP increases the synthesis of all nucleotides, whether it be purine or pyrimidine, through these two separate pathways. However, with purines, we're going to have it be the start of the synthesis pathway, whereas pyrimidines, it's going to increase its synthesis, but it's not actually going to conjoin with our nitrogenous ring until about halfway through formation, later on down here. When we look at purine anabolism, so the synthesis of purines, there are three main aspects that contribute to the synthesis. With de novo synthesis, guanine and adenine bases are actually synthesized from an intermediate. Both of these, though, can also go phosphorylation of these purines and phosphorylation of their nucleosides to regenerate them for use again. The amphibolic intermediate that we were looking at is IMP, inosine monophosphate, and so in our top corner up here, we're looking at our pentose phosphate molecule having a pyrophosphate being joined to that 2' carbon. And that pyrophosphate is very important to be there for the initial synthesis of the ring. And from here, we're going to go through a series of enzymatic reactions until we arrive at the amphibolic intermediate inosine monophosphate. And this construction occurs directly on the pentose phosphate ring, on the backbone up there. The double ring formation will occur directly on that pentose phosphate backbone. But once we have an inosine monophosphate, this will then undergo two different branches in order to either produce an adenosine monophosphate or guanosine monophosphate. And from there, it's either going to be converted with the enzyme adenylene succinate synthase or from IMP dehydrogenase, and this is going to be that split between either one. For guanine, its intermediate is going to be xanthosine monophosphate. For adenosine, we have adenylyl succinate, and so this is going to be its intermediate. Purines themselves, though, often go under salvage pathways. And this is going to involve two main options. So for that, we can reattach a free ring that doesn't have a phosphoribosylate sugar using adenine phosphoribosyltransferase or hypoxanthine guanine phosphoribosyltransferase. And in this case, the free ring is going to attach to the phosphoribosylate molecule, and it's going to release a pyrophosphate in the meantime. For our second one, if we already have the phosphate already on there, or if we're lacking the phosphate itself, but we have the sugar on there, the ribose sugar, then a phosphate can be added there using ATP. And so this is going to be our phosphorylation of a nucleoside. Now, for anabolisms of pyrimidines, it's a little bit different. We do also still have an amphibolic intermediate. This is going to be initiated from the generation of a molecule we call CAP, or carbamyl phosphate, which is composed of glutamine, carbon dioxide, and ATP. This is going to join with aspartic acid in order to create our ring, and through a dehydrogenase reaction, we are going to close that ring, and eventually we're going to blend up at uridine monophosphate. So after 6 different enzymatic reactions, we're going to get a uridine monophosphate, and this is going to be our intermediate as well. It is going to undergo phosphorylation to uridine diphosphate, but from there our path is going to split. If it undergoes deoxy, then it's eventually going to become a thymine monophosphate. Otherwise, it can be phosphorylated to uridine triphosphate, and then of course that can be converted using CTP synthase into a cytosine triphosphate molecule. Salvage pathways occur less frequently for pyrimidines. There's not as many opportunities or as many pathways for them to be salvaged from as you get with purines, though deoxycytidine, cytidine, and uridine can be phosphorylated back, and they can go from their NDPs to their NTPs. There is no direct pathway for the salvage of thymidine. However, the nucleosides deoxycytidine and deoxyuridine can be converted to thymidine, and so in this case we can somewhat salvage thymidine by converting other nucleosides that have been salvaged. But the synthesis of thymidine does require a tetrafolate to be put into the process. So because it requires tetrafolate and it requires additional steps for salvage, this molecule is really good at regulating and giving us our status for our nucleotides in general. So high concentrations thymidine diphosphate is actually used as a inhibition feedback for PRPP, which, going back to PRPP, we saw increases the synthesis of all nucleotides. Purines and pyrimidines, their catabolism here, this is the next phase we're going to be looking into. They're going to break down into 5 main byproducts. That's going to be uric acid, carbon dioxide, ammonia, beta-alanine, and beta-amino isobutyrate. And then I also wanted to add on there, we are going to have hydrogen peroxide generated during this. However, it's not going to be a direct byproduct from our breakdown of metabolism here. These byproducts may pass directly to the filter or filtered out through the kidneys, and then they may be utilized in cellular components. In the case of carbon dioxide, it can be exhaled via respiration. So that's typically how we're going to get rid of our waste products, is we're either going to use them in cellular components if need be, or the kidneys and the lungs are going to get rid of our waste products from the breakdown of purines and pyrimidines. For purines themselves, uric acid is the main byproduct of the degradation here. Adenosine and guanine are broken down to xanthine. This is actually, for adenosine, we use the enzyme adenosine deaminase, which will bring us back to our amphibolic intermediate inosine. Inosine and guanosine then will undergo a second enzymatic reaction called purine nucleoside phosphorylase, and this enzyme will get rid of our phospholipase backbone that they've been on. So now we'll be dealing with just the ring itself as the molecule will be degrading. And when it does that, we break down guanosine to guanine and then hypoxanthine to inosine to hypoxanthine. Guanine and hypoxanthine are going to both convert to xanthine, though through 2 different pathways. With hypoxanthine, we're going to add a water and oxygen molecule, and we are going to generate hydrogen peroxide. Whereas a guanine, we are going to remove an ammonia molecule, and from there we will have a xanthine again. Xanthine then will be converted to uric acid via xanthine oxidase. This is also going to intake water and oxygen and produce hydrogen peroxide. And so the generation of reactive oxygen species comes as a byproduct of the breakdown of purines here, even though their main output byproduct is going to be uric acid. With pyrimidines, it's a little bit different with their catabolism. We are going to have 4 main byproducts: carbon dioxide, ammonia, beta-alanine, and beta-amino isobutyrate. Beta-alanine, of course, belonging to cytosine and uracil, and beta-amino isobutyrate belonging to thymine during their breakdown. Cytosine actually first undergoes a reaction back to uracil, in which it is deaminated and oxygen is replaced, thereby creating a uracil. And then both uracil and thymidine are going to undergo enzymatic reaction with dihydropyrimidine dehydrogenase, and this is also going to require the input of NADPH. But this is essentially going to break down the double bonds on the molecule, which will then allow it to be free for cleavage by water. Once it has been cleaved, then the enzyme beta ureopropanase will then break them down into beta-alanine or beta-amino isobutyrate, depending on which one we're working with, and it will release also carbon dioxide and ammonia as waste byproducts. The next big things I wanted to look at was the biomedical importance of purine and pyrimidine metabolism. One of the big things that we notice with purine and pyrimidine metabolism is that these are nucleotides we're talking about, and so if we have any kind of disruption in DNA, then cancer can be a major issue that arises from this. And so their metabolism helps us find ways to fight cancer. Specifically, this quote from chapter 33 highlights this with purine deficiency states, while rare in humans, generally reflect a deficiency of folic acid. Compounds that inhibit formation of tetrahydrofolates and therefore block purine synthesis have been used in cancer chemotherapy. And here we're seeing at a list of some of the medications that are used. We also see with thiamine that because thiamine requires tetrafolate, a dihydrofolate compound, it is very sensitive to inhibitors of dihydrofolate reductase, and this can often also be used to treat cancer with. Lastly, we're going to look at our diseases that we are associated with purine and pyrimidine metabolism. And so there's two main arching branches that we're looking at here. With purine diseases, we see that we get hyperuricemia, Leishman syndrome, von Gert's disease, hyperuricemia. Gout is also placed there, as clinical defects of PRPP will present as gout. And since PRPP is the initial starting point for purine metabolism, having the ring being formed on there itself, that's associated with our purine metabolism here. With pyrimidine metabolism, one of the bigger things that we see is orotic acid, aciduria, both types 1 and type 2. Type 1 can affect our blood cells a little more heavily, creating megaloblastic anemia. So here we have 5 questions over the information that we have looked over. So I'm going to give you a couple of minutes to look over these questions, and then I'm going to go over their answers with you. And so our first question here, what is the intermediate that adenosine and guanine derive from? Back in purine anabolism, we saw the inosine monophosphate is our amphibolic intermediate through which both of these are derived from. Our second question here, phosphoribosyl pyrophosphate, PRPP, deficiency presents clinically as what disease? So in the previous slide, we were seeing that it is under purine metabolism, and when we have deficiency, it will clinically present as gout. Third question, what is the byproduct of purine catabolism? Our main byproduct of that is going to be uric acid. Of course, that then will be filtered out to the kidneys. And then which molecule is specifically required for thiamine production? That's going to be tetrafolate, being part of that dihydrofolate compound. Our last question here, hyperuricemia is associated with a deficiency of which enzyme? And that is going to be xanthine oxidase. And so we get hyperuricemia when we don't have the ability to break down xanthine. Thank you for watching my video and for learning about the metabolisms of purine and pyrimidines. I hope you enjoyed learning about the synthesizing and breaking down of nucleic acids.